fingerprinting. And we can see that there are certain exceptions to DNA fingerprinting. Exceptions to DNA fingerprinting means that a person can have more than one DNA fingerprint also. So what are the exceptions to DNA fingerprinting, and how can a person have more than one? Good morning, everybody. How can a person have more than one DNA fingerprint? Clear? So let's see that. And let's see a condition which is called as chimerism. See, human chimera. Last class you have studied that how DNA fingerprinting can be conducted. Human chimera is a person. I have talked about an example also last class, that on a US citizen a paternity test was conducted. He failed that test, and it was found that his son belongs to his uncle, but his uncle was never born. So this is an example of human chimera. So human chimera means what? Human chimera means that a person who would be having more than one DNA fingerprint, and that's exception to DNA fingerprint. So today let's study how is human chimera possible. How is human chimera possible? Clear? Now suppose we consider identical twins. Identical twins. Now when we talk about identical twins are genetically also same. Suppose this is an egg which has been fertilized by the sperm of father, and this results in the formation of a zygote, which would be obviously developing into a baby. Zygote, which obviously would be developing into a baby. But what happens? Sometimes division takes place in the zygote itself. And when division takes place in the zygote itself, two identical zygote would be formed. Two identical zygote would be formed when division takes place in the zygote itself. And these two identical zygote would be leading to the formation of what? Leading to the formation of two fetuses, which would be present inside the womb. But generally, they are present inside the womb in separate amniotic sac. Now, when we talk about the womb, we can see this is the mother's womb, and in the mother's womb, there is a sac which is called as what? Which is called as amniotic sac. Amniotic sac. Inside it, a fluid is present, and this fluid is called as what? It is called as amniotic fluid. This is a mother's womb in which a sac is present, which is called as amniotic sac, inside which the fluid is present, which is called as amniotic fluid, and this is fetus. This is fetus. Clear? This is the umbilical cord. Umbilical cord means the connect between mother and the baby, through which nourishment is also supplied to the baby, and this is called, also referred to as placenta. Umbilical cord would be called as placenta. Clear? And if suppose the mother feels some discomfort during pregnancy, a hollow needle would be inserted inside the womb. If the mother feels some discomfort during pregnancy, a hollow needle would be inserted inside the womb, and some of the amniotic fluid would be withdrawn. This is a hollow needle which would be inserted inside the womb, and some of the amniotic fluid would be withdrawn. Clear? Amniotic fluid would be withdrawn. Now, why amniotic fluid would be withdrawn? Because amniotic fluid consists of cells of the fetus. They consist of cells of the fetus, and these cells would be examined, that whether there is any kind of genetic disorder in the child or not. Clear? Whether there is any kind of genetic disorder in the child or not. So it would be examined. Fine. And if suppose there is a genetic disorder like Down syndrome, immediate step can be taken for its mitigation. we would be taking immediate step for its mitigation, or we'll say that please advise the parents not to go ahead with this child, abort that child, because if the child is born, he would be having trouble in future. So it's not good to have the child, abort the child. Clear? But this process is called as what? Through which we are responsible for withdrawing some amniotic fluid from the amniotic sac. This process through which we examine, because this fluid consists of cells of this fetus. Clear? So this process is called as what? It is called as amniocentesis. Sometimes it is known for wrong reasons also. Clear? But this is the way it is conducted. That a hollow needle is inserted inside the womb and some amniotic fluid would be withdrawn, and the fluid would be examined. The fluid would be examined, and what we can do is that the cells of the fetus would be present in that. That's why we say the fluid would be examined for any kind of genetic disorder, and if genetic disorder is present, then it would be advisable for the parents to abort that child. So this process is called as amniocentesis. Now generally what we can see in identical twins, the fetuses leading to the birth of identical twins would be present in the same amniotic, in different amniotic sac. But here we can see different amniotic sac. But sometimes in a rare case, it would be present in the same amniotic sac and share the same placenta. Now this type of identical twins, which is present in the same amniotic sac and is sharing the same placenta, is referred to as what? It is called as mono twins. It is referred to as mono twins. Clear? Mono twins. Same amniotic sac and share the same placenta would be called as mono twins. Now, what will happen? That sometimes what can happen? Now, this is the case of identical twin. Only we are talking. See, identical twins are identical. Sometimes the question is asked that whether the DNA fingerprint of identical twins would be same or not. Certainly, the DNA fingerprint would be same. They are genetically also same. The fertilization is one. Only division of zygote has taken place. So they are genetically same. Although some differences can creep in also due to positioning inside the womb, due to the amount of blood supplied. So that differences can creep in. Otherwise, genetically also they are the same. So this is about identical twins as such. Now, but you would have seen twins which are dissimilar in nature. They are not identical. These twins, now identical twins are also called as what? They are also referred to as monozygotic twins. But when twins are not similar, they are dissimilar, they would be called as what? They would be referred to as dizygotic twins. They are called as what? They are called as dizygotic twins. Dizygotic twins. This is called as what? This is called as mono twins. Not mono. Monozygotic twins. Monozygotic twins. And in this, a rare case where the fetuses leading to birth of identical twins would be present in the same amniotic sac and sharing the same placenta would be called as what? It would be called as mono twins. This is a case of mono twins. Clear? But if suppose the twins are not identical, you would have seen some twins which are not identical would be called as dizygotic twin, or would be called as fraternal twins. Fraternal twins. Take a screenshot of this. Take a screenshot of this. Fine. Now let's move. See how dizygotic twins or fraternal twins are born. See, fraternal twins which do not look similar, or we can see dizygotic twins. Dizygotic twins. dizygotic twins. Or dizygotic twins which do not look similar would be born. How? When the female mother is responsible for releasing more than one ovum at a time. Suppose she releases more than one ovum at a time, then these two ovums should be fertilized by the sperm of father. Clear? These two ovums should be fertilized by the sperm of father, and two zygote would be formed. Two zygote would be formed. But these are two different fertilizations. These are two different fertilizations, and they won't be similar. Definitely, they are not going to be similar because these are two different fertilizations. So, when they would be getting transformed into fetuses, and they would be also present in different amniotic sac, generally sharing the same and having different placenta. So, this is the dizygotic twins. So, when they are born, they do not have the same genetic makeup. And why they do not have the same genetic makeup? Because they have been born differently. They are fertilization has taken, two fertilization has taken place. In the case of monozygotic twins, they are similar. Why? Because only one fertilization has taken place. Clear? Only one fertilization has taken place. What has occurred is that the division has taken place in the zygote. Clear? Division has taken place in the zygote. Otherwise, only one fertilization, and that's why they are similar. In this case, they are not similar. Why? The genetic makeup is not similar. Why? Because this is the result of two different fertilizations. Me and my brothers, genetic makeup is not the same because we are the result of two different fertilizations. Clear? So they are just like, when they are born, they are just like brothers or sisters born at different periods of time. Clear? Brothers or sisters born at different periods of time. So this is called as what? This is referred to as the dizygotic twins or fraternal twins. Their features are not the same. You would have seen them that their features are not the same. But sometimes what happens? Sometimes what happens? The fetuses which are leading to the birth of fraternal twins are present in the same amniotic sac, and they are responsible for sharing the same placenta. They are present in the same amniotic sac, and they are responsible for sharing the same placenta. Then what happens? It means that this is A and this is B. The cells of A and B would be present in the amniotic fluid. Here is A, B, B. So cells of A and B are present in the amniotic fluid. Clear? Amniotic fluid. And when they are present in the amniotic fluid, what can happen during pregnancy is that cells of A would be absorbed by B and cells of B would be absorbed by A. Now, if this happens, then there would be two kinds of DNA fingerprinting present in these twins. One of their own and one of their fraternal brother or sister, as the case may be. Clear? So there would be two types of genome present in these persons, in these twins, one of their own and one of their fraternal brother or sister. Now, this is called as what? This process is called as what? This process is called as chimerism. When see, in this case, what is happening? The absorption of cell is responsible for multitude of genome in humans. If absorption of cell is responsible for multitude of genome in humans, then this process is called as what? This process is called as chimerism. Then this process would be called as chimerism. And the person would be called as what? The person would be called as human chimera. Because in this case, more than one genome is present in that person, more than one DNA fingerprinting is present in that person. So that person would be called as human chimera. And how this has happened? This has happened due to absorption of cell. So if absorption of cell is responsible for multitude of genomes in humans, this process is called as chimerism and the person would be called as human chimera. See, the if suppose division has taken place, why twins are always boy and boy, or girl, and why not boy and girl? See, fraternal twins can be boy and girl also. I told you dizygotic twins because it depends. See, in this case, when fertilization is taking place differently, so in when fertilization takes place, then only the sex is determined. Clear? In case of identical twins, the fertilization has already, the sex is itself determined. And then the division takes place in the zygote itself. Clear? The sex. But here, fertilization is responsible for determining of sex. Clear? So, two fertilization takes place. It's not that zygote fraternal twins cannot be boys and boys. They can be boys and boys, girls or girls also. But since two fertilization has taken place, that's why there's a possibility of a boy and a girl also. And it's not that boy or girl cannot take place. Boy or boy cannot take place. Girl or girl cannot take place. But in case of identical twins, it would be always of the same sex, because already fertilization has taken place. Only zygote division takes place after that, which results in. Clear? And Subham, it's not that it cannot be boys and boys. I have seen many fraternal twins which can be boys and boys, and girls. But sometimes, majority of the times, since they are of different fertilization, they can be boys or girls. No hard and fast rule for that. But then, in the case of identical twin, it would be always boy or boy and girl and girl. And the reason, because this is single fertilization. Only after fertilization, division in zygote has taken place. Clear? Here, there is no division in zygote. This is two different fertilizations. That's why the DNA fingerprint is different. Clear? Now, also, let's see where was the uncle, which example I cited. Sometimes what can happen? These two zygotes which have been formed, they can fuse with one another and develop as a single zygote. So here was the father who was born, here was the uncle who was not born. So uncle's zygote fused with that of the father, and father was born. So father had two kinds of cells, one of his own and one of his fraternal brother. So when the father reproduced, his reproductive cells had the DNA fingerprint of his own brother. That's why it was found that the child belongs to the uncle, but the uncle was never born. And why not born? Because the uncle's zygote fused with that of father. This is a type of chimerism also, where we can see the fusion takes place, and this is called as tetragametic chimerism. Why? Because four gametes, tetra means four. Four gametes have fused into one. Four gametes have fused into one. And sometimes what can happen, that after the birth of the child, the womb of the mother is cleaned. And the womb is cleaned as a precautionary measure, that no infection spreads inside the womb. But even after the cleaning of the womb, infections can be there. Even after cleaning of the womb, infections can be there. And because of this, what will happen? If there are infections inside the womb, because of this, what will happen is that infections will be there. And not only infections can be there, but also sometimes what can be there is the cells of the fetus can survive inside the womb. Clear? But it's properly cleaned so that no infection spreads. So the chances of infections are negligible. Sometimes it can be there. But the thrust is on our discussion that even after the cleaning of the womb, after the birth of the child, some cells of the fetus can survive inside the womb. And these cells would be traveling inside the mother's body and be absorbed by the mother's body herself, making mother human chimera, having more than one genetic makeup, one of her own and one of her son or daughter, as the case may be. Not son, but the daughter. Clear? So there can be possibility. Write down. Human chimera. Human chimera. Human chimera. If a person has if a person has more than one genetic makeup, if a person has more than one DNA fingerprint, sorry, if a person has more than one DNA fingerprint, he is referred to as human chimera. He is referred to as human chimera. Human chimera. It can take place under the following conditions. It can take place under the following conditions. First, when fetuses leading to the birth of when fetuses leading to the birth of fraternal twins leading to the birth of fraternal twins fetuses leading to the birth of fraternal twins are present in the same amniotic sac are present in the same amniotic sac sharing the same placenta are present in the same amniotic sac sharing the same placenta. It's a rare case. Exchange of cells between them exchange of cells between them exchange of cells between them can take place through amniotic fluid. Exchange of cells between them can take place through amniotic fluid. Which would be absorbed by each other's body. Which would be absorbed by each other's body. Absorbed by each other's body. See, I am not saying that both zygote of female are active in same time. I heard that something that female zygote active one by one. See, zygote means what? It's a fertilized egg. And when you see a number of babies born, you would have seen that ladies giving birth to more than one babies, 3 babies, 4 babies. How is that possible if there is no zygote at one time? First thing. Second thing is what? That zygote would be formed how? When a female is responsible for releasing more than one ovum at a time. So if suppose a female releases more than one ovum at a time in her cycle, which can be due to hormonal disturbance, etc., then only this is possible. Otherwise not. Generally, she is responsible for releasing one ovum at a time. Clear? But in exceptional cases, sometimes as an exception, due to hormonal disturbance, due to many other reasons, she would be responsible for releasing of more than one ovum at a time. Then this process would be occurring. When fetus, I repeat again, when fetuses leading to the birth of fraternal twins are present in the same amniotic sac and share the same placenta, exchange of cells between them exchange of cells between them can take place through amniotic fluid. Exchange of cells between them can take place through amniotic fluid. Which would be amniotic fluid which would be absorbed by each other's body. Which would be absorbed by each other's body. Resulting in more than one DNA fingerprint. And see, if absorption of cell is responsible for more than one DNA fingerprint, then the process is called as what? It is called as chimerism. And the person would be called as what? Human chimera. Write down. If absorption of cell is responsible for, if absorption of cell is responsible for more than one DNA fingerprint, if absorption of cell is responsible for more than one DNA fingerprint, then the process is called as, chimerism. Then the process is called as chimerism. Process is called as chimerism. Chimerism. And the person would be called as human chimera. Clear? But sometimes what can happen? Sometimes the two zygotes leading to the birth of fraternal twins can fuse with one another and develop as a single zygote. Clear? In this case also the person would be having more than one DNA fingerprint. That single zygote which would be developing into a baby. So a person would be having more than single DNA fingerprint, more than one DNA fingerprint. This process is called as tetragametic chimerism because 4 gametes have fused into one. Which type of twins is more common in nature? That's identical twins. Sometimes these zygotes leading to the birth of fraternal twins. So remember that it would be only possible in fraternal twins, not in identical twins, because in identical twins the genetic makeup is the same. Sometimes the zygotes leading to the birth of fraternal twins can fuse with one another, and develop as a single zygote, fuse with one another and develop as a single zygote, In this case also, now the person who would be resulting due to the single zygote, in this case also the person would have more than one DNA fingerprint. In this case also the person would have more than one DNA fingerprint. But this process is referred to as, tetragametic. But this process is referred to as tetragametic chimerism. But this process is referred to as tetragametic chimerism because 4 gametes have fused into one. And the third case is what? Sometimes the cells of the fetus can survive inside the womb. And if suppose the cells of the fetus can survive inside the womb, then what can happen is that it would be absorbed by the mother's body herself, resulting in more than one genetic makeup. And this process also would be leading to more than one genetic makeup or a chimera. Write down. Sometimes the cells of the fetus, can survive inside the womb after the birth of the child. Sometimes the cells of the fetus can survive inside the womb after the birth of the child. Clear? So they can survive inside the womb. And if this happens, they would be absorbed by the mother's body herself. Write down. These cells can be absorbed by the mother's body herself. Resulting in these cells can be absorbed by the mother's body herself, resulting in more than one genetic makeup for the mother herself. more than one genetic makeup for the mother herself. Mother herself. Is that clear? Fine. More than one genetic makeup for the mother herself. So these are the examples of human chimera, and this is how a person can have more than one DNA fingerprint. Now let's see the next topic. The next topic is called as what? It is referred to as stem cells. Stem cells. Now when we talk about stem cells, on stem cells they have asked you a number of questions. So let's see first of all, what exactly is the meaning of stem cell? What is stem cell? To understand this, let us understand first of all what is called as potency. See, when we talk about potency of a cell, potency of a cell. So potency of a cell is what? It's the characteristic through which a cell can convert into another type of cell. So we say that potency of a cell is the characteristic, through which it converts into another type of cell, that is called as potency. Clear? So it's a characteristic of a cell through which it converts into another type of cell. And there are different forms of potency. For example, suppose here we take, this is suppose the zygote which has resulted after fertilization. This is the zygote which has resulted after fertilization. Now after the formation of zygote, that is when fertilization has occurred, this zygote has been formed. We can notice a stage, and this stage is called as what? This stage is called as totipotent morula. So zygote is a single cell. How can a single cell develop into a fetus having many cells? Clear? Having many cells. Fine, thank you. And we can see that in the later stage of zygote formation, some cells would be coming into existence. 8 to 12 cells, let's take 9, comes into is formed, and this stage is called as what? It is called as totipotent morula. This stage would be referred to as totipotent morula. The characteristic of these cells is called as what? It is called as totipotency. Characteristic of these cells is called as totipotency. Toti means what? Toti means total. Potency means what? Potency means the ability to transform. Ability to transform. So we can see that these cells have the ability to transform into total types of cell, that is all type of cells. So this characteristic would be called as what? This characteristic would be called as totipotency. These cells have the ability to transform into all types of cell or total types of cell. That's why this characteristic would be called as totipotency. But in the later state of zygote formation, after this phase, we can see a phase is reached, and this phase is called as what? It is referred to as blastocyst. Now blastocyst means that in this phase there are a number of cells. Clear? A number of cells have been formed, and all these cells would be having a characteristic which is called as what? Which is referred to as pluripotency. So these cells would be having those characteristics which is referred to as pluripotency. Pluripotency means, pluri means many, Potency means the ability to transform. So these cells have the ability to transform into many type of cells, but not all. So these, here we have said, these cells have the ability to transform into total type of cell, all type of cell. These cells have the ability to transform into many type of cells, but not all, not total type of cells. Clear? So this would be called as what? This would be called as pluripotency. Fine. And you can see that these pluripotent cells, suppose these are the pluripotent cells, they would be developing into your respiratory system. They would be developing into your muscular system. And the baby would be formed. But this is called as pluripotency. This characteristic is called as pluripotency. So pluripotency means if the cell have a characteristic which is called as pluripotency, then it can transform into many type of cell, but it cannot transform into all type of cell. If a cell has a characteristic which is called as totipotency, it means that it can transform into all type of cell or total type of cell. Obviously, there would be a difference between totipotency and pluripotency. So the difference between pluripotency and totipotency are that of placental cells. You have seen the placental cells, the umbilical cord. So placental cell, it means that totipotent cells can transform into placental cells, but pluripotent cells cannot transform into placental cells. Clear? Pluripotent cells cannot transform into placental cell. See, if we are going to extract artificially one cell from the blastocyst stage, this stage, one cell is extracted. This is pluripotent cells. But if we take out one cell from this, the entire fetus is going to die. Why? Because all these cells are intertwined, all these cells are linked with each other. The entire fetus is going to die. So it won't be proper to extract pluripotent cells from the blastocyst stage. Clear? But when we talk about stem cells are those cells which are having a characteristic which is called as pluripotency. Clear? See, all these cells, the cells of the blastocyst stage, some of them would be developing into respiratory system, some the muscular system, some other systems of our body. But in this process, some cells inside human body would be left behind. They won't transform into other type of cells. And they are present in our developed body in the bone marrow. They are present in the developed body in the dental pulp. They are present in the developed body, not only in the bone marrow and in the dental pulp. They are present in the outer layer of the heart, and they would be called as somatic stem cells. Somatic stem cells. So why is that? Some of the pluripotent cells have developed into different types of organs, different types of cells of the organs. But why some were left behind? Now these some cells were left behind inside our body. They haven't developed because these cells have a gene present in them. And these genes do not allow them to undergo cell division of their own. So they haven't developed into different types of cell. This gene is called as nanog. So it means that stem cells also have a characteristic which is called as have a characteristic that they do not undergo cell division of their own. Because a gene is present in them, and that is called as nanog, which disallows it to undergo cell division of its own. Clear? Which disallows it to undergo cell division of its own. So that is called as nanog. First of all, write down potency of a cell, and then we write down the definition of stem cell, that what is exactly stem cell. Write down potency of a cell. Potency of a cell is the characteristic. Potency of a cell is the characteristic is the characteristic through which it transforms into. Potency of a cell is the characteristic through which it transforms into. Through which it transforms into different types of cells. Through which it transforms into another type of cell. Transforms into another type of cell. There are different types of potency. There are different types of potency. Different types of potency. Potency. First, totipotency. 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 This is the characteristic of the cell. This is the characteristic of the cell. Characteristic of the cell through which it can transform into. This is the characteristic of the cell through which it can transform into. It can transform into all, that is total, all or total type of cells, through which it can transform into all or total type of cells. All or total type of cells. Total type of cells. So that's called as what? That's called as totipotency. All or total type of cells. Total type of cells. For example, cells of the totipotent marula stage. Marula stage, I have written marula here, it should be here. Have this characteristic. Write down pluripotency. Pluripotency. It is the characteristic of the cell. It is the characteristic of the cell. Characteristic of the cell through which it can transform into. It is the characteristic of the cell through which it can transform into. Through which it can transform into. It can transform into only many type of cells. Through which it can transform into many type of cells. Many type of cells, but not all. Through which it can transform into many type of cells, but not all. But not all. Clear? Many type of cells, but not all. Fine. So this is called as what? Just 1 second. This is called as pluripotency. This is referred to as pluripotency. Fine. Referred to as pluripotency. Fine. Now, the difference between totipotency and pluripotency is that of the placental cells. So, totipotent cells can transform into placental cells, but pluripotent cells cannot transform into placental cells. Write down the difference between totipotency and pluripotency. The difference between totipotency and pluripotency. Totipotency and pluripotency is of placental cells. Is of placental cells. Totipotent cells can transform into placental cells. Totipotent cells can transform into placental cells. Totipotent cells can transform into placental cells, but pluripotent cannot. Totipotent cells can transform into placental cells, but pluripotent cannot. But pluripotent cannot. Clear? So they can transform into placental cells, but we can say pluripotent cells cannot transform into placental cells. So that's the difference. Fine. Transform into placental cells. So that's the difference between totipotency and pluripotency. Now come to the definition of stem cells. Write down, stem cells are those cells. Stem cells are those cells which are constantly present, which are constantly present. Stem cells. Take a screenshot of this. Fine. Stem cells are those cells which are constantly present, which are constantly present. Stem cells are those cells which are constantly present in their youthful stage, in their youthful stage, youthful stage. And why we are saying that they are present in their youthful stage? They are present in their youthful stage because they do not undergo cell division of their own. Write down as they do not undergo cell division of their own, as they do not undergo cell division of their own, do not undergo cell division of their own. They are said to be present in quiescent stage. They are said to be present in quiescent stage. They are said to be present in quiescent stage. Said to be present in quiescent stage. And quiescent stage means that they cannot undergo cell division of their own. So the meaning of quiescent stage is what? They do not undergo cell division of their own. Undergo cell division. They would be undergoing cell division if artificially chemicals are given to them, but they do not undergo cell division of their own. So we can see youthful stage, then quiescent stage also. So they are present in the youthful stage, and that means that they do not undergo cell division of their own. So if a cell is constantly present in youthful stage, it does not undergo cell division of its own, then this stage would be called as what? This stage is referred to as quiescent stage. Clear? This stage would be referred to as quiescent stage. And as far as the other characteristic of the stem cell is concerned, what are the other characteristic of the stem cells? We can see that why they are not undergoing cell division of their own? They do not undergo cell division of their own because a gene is present in them which is called as nanog. Write down. They do not undergo cell division of their own. They do not undergo cell division of their own as a gene is present in them, as a gene is present in them called nanog, as a gene is present in them called nanog. So nanog disallows it to undergo cell division of its own. Clear? This gene which is called as nanog would be disallowing it to undergo cell division of its own. Clear? So this is, and you can see similar characteristic is present in viruses also. Viruses also do not replicate of their own, and they do not replicate of their own. Why? Because critical genes are present in viruses for this purpose. So the characteristic of stem cell and that of the viruses happen to be very similar. Clear? The characteristic of stem cell and that of the viruses happen to be very similar. So gene is present in them which is called as nanog, which disallows it to undergo cell division of its own. But see, if chemical instructions are given to them artificially, they would be undergoing cell division. They would be transforming into different types of cell. Write down. If chemical instructions are given to them, if chemical instructions are given to them, instructions are given to them, instructions are given to them, they can transform into different types of cells. If chemical instructions are given to them, they can transform into different types of cell, different types of cells. Clear? And this process through which chemical instructions are given to them, they can transform into different types of cells. This process can be utilized for making of organs, can be utilized for making of not only organs, but also can be utilized for making of organs, and also utilized for making of not only organs, but also curing diseases, tissues. Write down. This process, this process is referred to as, this referred to as means when chemical instructions are given to these cells and they transform into another type of cell. This process is referred to as differentiation. 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 This process is referred to as differentiation. Clear? And we can see that by this process, stem cells can be used for what? It can be used for development of tissues, and it can be used for development of organs. It can be used for curing diseases. And this characteristic of this stem cell is very well known to us. Write down. By this process, organs can be developed. By this process, organs can be developed. Organs can be developed. Tissues can be formed. Tissues can be formed, and diseases can be cured. And diseases can be cured. Diseases can be cured. Clear? So, as a conclusion, we can say that if you can develop tissues, if you can cure diseases, if you can develop organs, then with the help of the stem cells, then it means that stem cells have vast curative potential. And since stem cells have vast curative potentials, they are having vast curative potentials. That's why they are called as master cells. Write down. Stem cells have vast curative potentials. Stem cells have vast curative potential. Stem cells have vast curative potential. And since they are having vast curative potential, that's why we call them as what? We are also referring them to as master cells. Write down. As such, they are also referred to as such, they are also referred to as master cells. As such, they are also referred to as master cells. Master cells. Clear? So you can see all the points are there before you, that stem cells are those cells which are present in the youthful stage. And youthful stage, why? Because they do not undergo cell division of their own. Stem cells are those cells which are present in quiescent stage. Quiescent stage means that if a cell does not undergo cell division of its own, then this stage is called as what? This stage would be referred to as quiescent stage. Clear? And why they are not undergoing cell division of their own? Why? Pluripotent cells, they have a now see, nano is a gene which is present in them. Now this gene disallows it to undergo cell division of its own. Nano is a gene which is present in them, and this gene disallows it to undergo cell division of their own. Clear? Of its own. And this gene is simply disallowing it. So you can see characteristic of stem cell is very akin to, very similar to viruses, because viruses also do not undergo do not replicate of their own. They replicate only inside the host. So viruses also do not replicate of their own. They replicate only inside the host. And but if chemical instructions are given to these cells, artificially if we are responsible for giving them chemical instructions, then these cells can transform into different types of cells. Clear? Then these cells would be transforming into different types of cell. This process is called as differentiation. Differentiation means that the process through which you are giving chemical instructions to them so that they transform into different types of cells. And this process can be used for making of organs, tissues, diseases. Today what happens? That if an organ becomes dysfunctional, stem cell can be used for making that organ again inside the body also. Clear? So organs, tissues, and diseases would be cured. So you can see that stem cells have vast curative potential. Vast curative potential. And since they are having vast curative potential, that's why we are referring them to as master cells. So take a screenshot of all these points, which you have written also, but in a abridged form, it is like this. They are also called as master cells. And do remember that the first stem cell transplant center in India was established at Chennai. The first stem cell transplant center in India was established at Chennai. So this is about stem cell, the introductory part of this. Clear? Take a screenshot of this. Now, let's deal with the types of stem cells. That what are the types of stem cells? So let's cover up that. So suppose we take let's consider the types of stem cell. We can see that stem cells can be broadly divided into two types. Stem cells can be broadly divided into two types. Now, first type is called as what it is called as embryonic stem cell. So this is one type which is called as embryonic stem cells. Clear? Embryonic stem cells. That's first type of stem cell. And embryonic stem cell means what? Embryonic stem cell means that this type of stem cell is extracted from initial phase of life. This type of stem cell is extracted from initial phase of life. For example, it is extracted from the blastocyst stage. It would be called as embryonic. But you cannot extract stem cell from the blastocyst stage because this would be unethical in nature. Why? Because if you take a single pluripotent cell from there, the death of the fetus would take place. So this would be unethical in nature. So we won't be doing that. Clear? So we cannot extract stem cells from the blastocyst stage. But these are what embryonic stem cells are, those stem cells which are present in the initial phase of life. Embryonic stem cells are those stem cells which can be extracted from initial phase of life. So the first point we'll write about this is that they are extracted from initial, they are extracted from initial phase of life. Initial phase of life. That means blastocyst stage also. They are extracted from initial phase of life. That's the first thing. Clear? So they are extracted from initial phase of life. One thing that we can talk about first. But it means that also that the sources of this embryonic stem cell can be what? The source can be blastocyst stage. So if suppose we talk about the sources, that what are the sources of embryonic stem cell? The sources would be blastocyst stage. But then when you write blastocyst stage, you would be striking it off also. Why? Because extracting embryonic stem cells from the blastocyst stage would be considered as what? It would be considered as unethical in nature. Okay, I'm coming to it. Why it is said. Then from where we should be extracting embryonic stem cell? The answer to your question is here only. We can see after the birth of the child, inside the womb, what is left? Amniotic fluid, peripheral blood, and umbilical cord. Now this waste would be taken out of the womb. This is the waste. But it's also, on the other hand, a rich source of embryonic stem cell. So if we cannot extract embryonic stem cell from blastocyst stage, from where we would be extracting. From here we would be extracting. So the sources also includes what? The sources also includes umbilical cord. The sources also includes amniotic fluid. The sources also includes not only umbilical cord, amniotic fluid, but also peripheral blood. Clear? Peripheral blood. And we can see that the cells extracted from here, umbilical cord, amniotic fluid, peripheral blood. Now this is nothing but embryonic stem cell. Now these would be stored where? It would be stored in a stem cell bank. Stem cell bank. I hope the question that you have asked is clear, that many a times it is said to preserve the umbilical cord, umbilical fluid at the time of the birth. Why? Now this would be stored in a stem cell bank, and if suppose the child meets an accident in future, these cells can be utilized for making of organs or curing diseases or making of tissues of that child. Clear? Making of tissues of that child. So this is embryonic stem cell. But the second type of stem cell would be called as what? It would be referred to as somatic stem cell. So we would be calling that as somatic stem cells. And when we talk about somatic stem cells, somatic stem cells are present inside the developed body. Clear? Somatic stem cells are present inside the developed body. And just one second. When you're talking about somatic stem cells, we can see somatic stem cells are present inside the developed body. And why somatic stem cells? And what would be the source of somatic stem cells? Somatic stem cells, we say, are present inside the developed body. So what would be the source of somatic stem cells? What are the sources? And these are present inside the developed body. So you can write like this: developed body, present inside the developed body. And the sources of somatic stem cell include what? It includes bone marrow. It includes dental pulp. It includes outer layer of heart. Outer layer of heart. So these are the sources of somatic stem cell. But the question is that if we have somatic stem cells in the body, then why should we be responsible for extracting embryonic stem cells? Embryonic stem cells would be extracted. Why? Because the differentiation ability of embryonic stem cell is far more than compared with somatic. So you can see somatic stem cells are present inside the developed body. The sources include bone marrow, dental pulp, outer layer of the heart. Take a screenshot of this. Just I am coming to that. Cancer stem cell. Fine. Now the question is that somatic stem cells and embryon. If somatic stem cells are present in our body, why is embryonic stem cell also kept stored in a stem cell bank? Write down. Embryonic stem cells are preferred over somatic stem cells. Embryonic stem cells are preferred over somatic stem cells. Somatic stem cells. As the differentiation ability of as the differentiation ability of somatic stem cell is very high. As the differentiation ability of somatic stem cell is very high. Somatic stem cell is very high. Differentiation ability of not somatic cell. Embryonic stem cell is very high, as the differentiation ability of embryonic stem cell is very high when compared to somatic. That's why in stem cell therapy, embryonic stem cell is preferred over somatic stem cell. In stem cell therapy, embryonic stem cell is preferred over somatic stem cell in stem cell therapy, as the differentiation ability of embryonic stem cell is very high. Clear? Differentiation ability of embryonic stem cell is very high. But there are two other types of stem cell that you have to know. One type of stem cell is called as what? It is called as iPS cells. iPS cells means induced pluripotent stem cells. Induced pluripotent stem cells. Induced pluripotent stem cell. So this is called as what? This is called as iPS cells. And what is iPS cells? See, viruses have characteristic which is very akin to stem cells, because viruses also do not replicate of their own. And just like stem cell, in viruses, we can see a gene is present. And this gene is called as what? This gene is referred to as critical genes. So this gene is called as critical gene, which is present in viruses. So just like stem cell, a gene is present which is called as critical gene, and that is present in viruses. Clear? That is present in viruses. So two scientists, what they did was, they took somatic cells. Suppose these are somatic cells. Our body is divided into two cells. One is your germline cells, that is called as reproductive cells. And barring all germline cells, barring the reproductive cells, the cells which are present inside your body, barring reproductive cell, the cells which are present inside your body is called as what? It is called as the somatic cells. So somatic cells you can say, or you can say body cells. So these are cells which are except the reproductive cell, all the cells which are present inside our body would be called as body cells or somatic cells. Now, two scientists, they took what? They took, and their name was Gordon and Yamanaka. They took somatic cells, and not somatic stem cells, somatic cells, and exposed them in front of viruses, containing critical genes. And you know, critical genes are very similar to NANOG. And some chemical instructions were given to them, to these somatic cells. So when they were exposed in front of viruses containing critical genes, what happened was, automatically the critical genes were absorbed by these cells. Automatically the critical genes were absorbed by these cells, and they transform into pluripotent stem cell. The somatic cells, they transform into pluripotent cells. So they are called as what? They are called as, since the mechanism is what? Mechanism is artificial. So since the mechanism is artificial, they are referred to as what? They are referred to as iPS cells, induced pluripotent stem cells, and they can be used for stem cell therapy. Clear? They can be used for development of organs. They can be used for development of tissues. So this can be done. And for this purpose, the two eminent scientists, Gordon and Yamanaka, they were conferred Nobel Prize in the field of medicine. Gordon and Yamanaka were conferred Nobel Prize in the field of medicine. Fine. So this was done. Write down. Induced pluripotent stem cell. When two scientists, Gordon and Yamanaka, Gordon and Yamanaka, when two scientists, Gordon and Yamanaka, exposed some somatic cells, exposed some somatic cells, some somatic cells, exposed some somatic cells in front of viruses containing critical genes, in front of viruses containing critical genes, viruses containing critical genes, in front of viruses containing critical genes. And chemical instructions were given to these cells. And chemical instructions were given to these cells. Chemical instructions were given to these cells. And chemical instructions were given to these cells. They absorbed these cells. They absorbed the critical genes, and chemical instructions were given to somatic cells. Make it specific. And chemical instructions were given to somatic cells. Somatic cells. They absorb the critical genes. They absorb the critical genes and transformed into pluripotent cells. And transformed into pluripotent cells. They absorb these genes and transformed into pluripotent cells. Transformed into pluripotent cells. Pluripotent cells. Clear? So this process is called as what? This process is called as differentiation. Write down. This process, transform into pluripotent cells, and this can be used for differentiation. But just hold on. That these cells, they absorb these genes and transform into what? Pluripotent cells. And this can be used for differentiation means that this can be used for stem cell therapy. Write down. These somatic cells can be used for stem cell therapy. These somatic cells can be, because they are just like stem cells. These somatic cells can be used for stem cell therapy. Somatic cell can be used for stem cell therapy. These somatic cell can be used for stem cell therapy. Used for stem cell therapy. Clear? So they can be used for stem cell therapy. Fine. Stem cell therapy. They can be used for stem cell therapy. Fine. Now these are called as what? These are called as induced pluripotent stem cells. Right? And they are referred to as, because the mechanism is artificial, they are referred to as induced pluripotent stem cells. They are referred to as induced pluripotent stem cells. Induced pluripotent stem cells. Induced pluripotent stem cells. Clear? They are referred to as induced pluripotent stem cells. Clear? And they can be used for, and for this purpose, Goodon and Yamanaka were provided the Nobel Prize in the field of medicine for the year 2012. Right down. For this purpose, Goodon and Yamanaka, for this purpose, Goodon and Yamanaka were conferred. Goodon and Yamanaka were conferred Nobel Prize in the field of, were conferred Nobel Prize in the field of, Nobel Prize in the field of, in the field of medicine, in the field of medicine in the year 2012. In the year 2012. Now the last stem cell, and that is called as what? That is called as cancer stem cell. See, when you talk about cancerous tumors are what? Cancerous tumors are malignant. Malignant means they continuously grow. So cancerous tumors are malignant in nature. They continuously grow. So that's cancerous tumors. But and the early perception was all the cells within a cancerous tumor have the ability to replicate, to differentiate at a very high speed. That's why we say that all these cells in a cancerous tumor have the ability to replicate or to differentiate at a very high speed. Clear? So that's about cancer stem cell. But now the perception is what? Now the perception is only few cells have this ability to differentiate at a very high speed. Only few cells have the ability to differentiate at a very high speed. And due to their high differentiation ability, they are referred to as cancer stem cells. Clear? Now, the problem with cancer patient is what? That when chemotherapy is applied for the cancer patient, and obviously you know that chemotherapy is done for the cancer patient. So, when chemotherapy is applied for the cancer patient, the purpose is what? The purpose is to destroy the cancer stem cells, but it results in destroying the normal somatic stem cells which are present inside the body. Clear? It results in destroying the normal somatic stem cells which are present inside the body. So that's the problem with cancer stem cells. Clear? So this is called as cancer stem cells. Write down cancer stem cells. Cancer stem cells. Inside cancerous tumors. Inside cancerous tumors. Cancerous tumors. Only few cells have the ability. Only few cells have this ability. Have the ability. Have the ability to differentiate at a very high speed. Only few cells have the ability to differentiate at a very high speed. To differentiate at a very high speed. Only few cells have the ability to differentiate at a very high speed. Due to their high differentiation ability. Due to their high differentiation ability. High differentiation ability. Due to their high differentiation ability, they are referred to as cancer stem cells. They are referred to as cancer stem cells. Cancer stem cells. Cancer stem cells. When chemotherapy is applied, chemotherapy is applied, the purpose is to destroy cancer stem cells. When chemotherapy is applied, the purpose is to destroy cancer stem cells. Purpose is to destroy cancer stem cells. But it results in destroying. It results in destroying the normal somatic stem cells present inside the body. Normal somatic stem cells present inside the body. But it results in destroying the normal somatic stem cells present inside the body. Stem cells present inside the body. Normal somatic stem cells present inside the body. Clear? So it results in destroying that. The normal somatic stem cells present inside the body. Normal somatic stem cells present inside the body. Fine. Clear? So this is about cancer stem cell that you have to know. But for this purpose, what can be done? If suppose cancerous tumor is there, Now for this purpose we can use, there is a cancerous tumor and you have to destroy this tumor. You don't have to destroy the other cells nearby. So for destroying cancerous tumor we can use a device which is called as cyber knife. Cyber knife has two parts. One is called as what? One is called as linear particle accelerator. Linear particle accelerator. And this is for the purpose of developing radiation. And second part is called as what? Second part is called as robotic arm. Now this is for the purpose of directing the radiation towards the cancerous tumor in exact precise dose, so that the cancerous tumors gets destroyed but the cells nearby are not affected. Clear? So this is called as what? This is called as cyber knife. Write down. For cancerous tumors, a robotic device called cyber knife can be used. A robotic device called cyber knife can be used. A robotic device called cyber knife can be used. Cyber knife can be used. Clear? It has two parts. First, linear particle accelerator. It has two parts. First, linear particle accelerator. Two parts. First, linear particle accelerator. And second, robotic arm. Robotic arm. Clear? Now, the first part, that is linear particle accelerator, is responsible for developing the radiation. Right? Now, the first part is responsible for developing the radiation. The first part is responsible for developing the radiation. And the second part, robotic arm. The first part is responsible for developing the radiation, and the second part, robotic arm. First part is responsible for developing the radiation. Second part, robotic arm, directs the radiation in exact precise dose. Directs the radiation in exact precise dose. That is, the amount of radiation which is required would be directed in exact precise dose. Over the cancerous tumor. In exact precise dose over the cancerous tumor. So that the tumor gets destroyed. But cells nearby. Are not affected. Now this is about stem cell that you have to know. What is stem cell? What is potency? And then what is a stem cell? What is differentiation? Two types of stem cell, embryonic, somatic. And then we can see iPS cells. Then what is called as cancer stem cells, and also what is cyber knife. Now, the next topic that we have to cover up is called as what? It is called as cloning. Clear? So let's cover up the next topic, and that topic is called as cloning. So what is cloning? See, as far as cloning is concerned, we can see the first successful experiment of cloning was conducted in 1997. And was conducted in 1997 when? A sheep called Dolly was born, and Dolly was born at Roslin Institute, Edinburgh. Edinburgh is in Scotland. So a sheep called Dolly was born. So it was cloned. Dolly was born. Now it was born under the supervision of Dr. Ian Wilmot. And cloning is a mechanism. When we define cloning, it is a mechanism in which identical replica of an organism would be prepared. Suppose myself, same myself, just like me, other person would be prepared, would be appearing to be just like the same. So he would be physically and genetically same to me. So my clone as such, physically also, genetically also same. So we define cloning as what? It is a process through which identical replica of an organism would be prepared, which is physically and chemically, not chemically, physically and genetically similar to the donor. Physically and genetically similar to the donor. So that's the definition of clone. And this mechanism is based on what? This mechanism is based on somatic cell nucleus transfer. So let's see how it is based on somatic cell nucleus transfer and how cloning would be conducted. So how cloning is conducted? Let's see. Let's see. On one side, we are responsible for taking the egg provider. Egg provider means the person who would be responsible for providing the egg. So on one side, we are taking the egg provider. Clear? So C has been taken. And on the other side, we are taking the donor. On the other side, we are responsible for taking the donor. The donor. Donor is a person whose clone is to be prepared. So now from the donor, we are responsible for extracting some somatic cells. Somatic cells would be not somatic stem cells. Somatic cells would be extracted. So let's extract some somatic cells. And these somatic cells would be extracted from the donor through surgery. It's not that we would be squeezing it from out of the body of the donor. So we would be extracting these somatic cells through surgery. Clear? So when it's extracted through surgery, it would be placed in a quiescent stage. Quiescent stage means that in this stage, no cell division takes place. So it would be placed in quiescent stage. Quiescent stage. Clear? Now from the egg provider, quiescent stage here means that we are responsible for when the cell is extracted, somatic cell is extracted from the body, then cell division stops. So we are maintaining that same stage. Clear? That cell division stops, and this stage in which cell division stops would be called as quiescent stage. Now what we are doing is that we are responsible for giving these cells chemical instructions. Chemical instructions are given to these cells. And the purpose of giving them chemical instruction is what? So that they return back in their life cycle. See, cells are born, cells mature also, they die off also. But here we are responsible for giving them chemicals so that they revert back in their life cycle, so that they go back in their life cycle. Clear? And there are some chemicals which can be reverting these cells back into their life cycle. And after this, the cell, suppose, which has reverted back in its life cycle to the utmost, would be selected. This cell would be selected. Clear? So one somatic cell has been selected, which has been reverted back into its life cycle. On the other hand, from the egg provider, we extract the egg. and here is the nucleus. So first of all, what we are doing is that we are responsible for removing the nucleus from the egg cell. So we have removed the nucleus from the egg cell and it has become enucleated. So it is devoid of nucleus, any kind of nucleus. Clear? This cell would be now brought here. And what will happen? That the nucleus of the egg cell, nucleus of the egg cell, this is the nucleus. Now this would be transformed inside the, this would be transferred, not transformed, this would be transferred to the egg cell. Now this mechanism of transferring, the nucleus of the somatic cell to the egg cell is called as what? It is called as SCNT. SCNT means somatic cell nucleus transfer. Somatic cell nucleus transfer. Clear? Somatic cell nucleus transfer. And then we can see that this would be now planted inside the womb of mother, surrogate mother, egg. The baby which is born would be a clone of donor. I repeat. Now from one side, on one hand we have taken the egg provider and from her we have extracted one egg. So this is the egg that we have extracted, and we mechanically the nucleus would be removed from it. Mechanically it can be removed. See, it is not grown in the lab with the help of embryonic stem cell. It is grown in the lab with the help of tissue culture. And that meat I have taught you earlier also, which is called as mosa meat, when I completed with you ruminants as a contributor to global warming. Clear, Nirmal? So first of all, we are responsible for extracting the egg from the egg provider. Then mechanically we are responsible for removing the nucleus from it. It becomes enucleated. On the other hand, we have taken the donor. From the donor, we have taken through surgery some somatic cells. Now, some somatic cells, and these somatic cells would be put in the quiescent stage. Quiescent stage means that the cell division would be stopping in this stage, and then we are responsible for giving them chemical instructions so that they revert back into their life cycle. One cell would be selected and the nucleus of this cell would be transferred to the egg where the nucleus has been removed, and this would be done with a spark of electricity also. Spark of electricity, so that the egg feels that it has been fertilized, but although no fertilization takes place in cloning, and this would be now transferred to the womb of surrogate mother. The baby that is born would be a clone of the donor. The baby that is born is clone of the donor, but I say it won't be exactly the same. And tell me why it won't be exactly the same. Tell me why it won't be exactly the same. Although it is a clone, but it cannot be considered as 100% exact. Why it cannot be 100% exact? The real reason is what? That mitochondria has not been transferred. Clear? Mitochondria is present here. Mitochondria is present here. Mitochondria is present here. So mitochondria is still belonging to whom? It is belonging to the egg provider. Clear? So it has not been transferred. So it cannot be called as exactly the same. But it can be exactly the same if the egg provider and the donor happens to be the same person. If the egg provider and the donor happens to be the same person, then it can be exactly the same. So it means what? It means that girl child can be an exact clone of the mother, But boy child cannot be an exact clone of the father because mitochondria is not the same. Is that clear? Mitochondria is not the same. Now, this mechanism of cloning, this method of cloning that we have discussed is referred to as what? It is called as reproductive cloning. This method, reproductive cloning. This method of cloning is called as what? It is called as reproductive cloning. Fine, reproductive cloning. But there's another method of cloning also. And let's see that method also. Write down cloning. Take a pic of this first of all. Take a pic, screenshot of this. Write down cloning is a mechanism. Cloning is a mechanism. Is a mechanism with the help of which, cloning is a mechanism with the help of which identical replica of an organism is prepared. Cloning is a mechanism with the help of which identical replica of an organism is prepared. Identical replica of an organism is prepared. Which is genetically and physically similar to the donor. Which is genetically and physically similar to the donor. Genetically and physically similar to the donor. Genetically and physically similar to the donor. Similar to the donor. Clear? The first successful experiment of cloning. The first successful experiment of cloning. Was conducted in 1997. The first successful experiment of cloning was conducted in 1997. Was conducted in 1997. 1997. Conducted in 1997. When? Conducted in 1997. When a sheep called Dolly, was born. A sheep called Dolly was born. At Roslin Institute. Edinburgh. At Roslin Institute, Edinburgh. Edinburgh is in Scotland. Roslin Institute, Edinburgh. Clear? And cloning is based on what? Cloning is based on somatic cell nucleus transfer. Write down. Cloning is based on somatic cell nucleus transfer. Somatic cell nucleus transfer. And see why we have reverted this back into its life cycle, because all the earlier experiment of cloning which was conducted, which resulted in a failure, that was conducted. In all those experiments, what we did was we directly transferred the nucleus from the somatic cell to the egg without reverting this back into their life cycle. If you compare somatic cell with the egg, we can see that somatic cells and egg are in two different phases of their life cycle. Egg is in the incipient phase, beginning phase. So egg is in incipient phase or beginning phase. Clear? And as far as somatic cell is concerned, somatic cell is a developed phase. So if we are going to transfer nucleus from a developed cell to a cell which is in its incipient stage, which is in its beginning phase, certainly what will happen is that nucleus transfer in this case won't be compatible. Clear? Nucleus transfer in this case won't be compatible. So what we do is we are responsible for reverting these cells back in their life cycle so as to make nucleus transfer more compatible. Clear? So as to make nucleus transfer more compatible. So that's why we are responsible for reverting these back into their life cycle. So that's the reason why we reverted it back into the life cycle. Clear? Now, what is the mechanism of pruning? So it is based on somatic cell nucleus transfer. Right on one side, from the egg provider, on one side from the egg provider, egg is extracted, and mechanically, nucleus is removed from it, nucleus is removed from it. Clear? So it becomes what? It becomes enucleated. It is devoid of nucleus. Right. On the other hand, from the donor, somatic cells are extracted from the donor. Somatic cells are extracted, and they are put in a quiescent stage. Quiescent stage means cell division stops, and they are put in a quiescent stage. Now the real thing happens here. Chemical instructions are given to these cells so that they revert back in their life cycle. Right down. Chemical instructions are given to these cells. So that they revert back in their life cycle. Chemical instructions are given to these cells so that they revert back in their life cycle. So they go back in their life cycle. And after this, we can see one cell is selected, and the nucleus of this cell would be transferred to the egg with a spark of electricity. Right down. After this, one cell is selected. And the nucleus of it is transferred. One cell is selected, and the nucleus of it is transferred to the egg. With a spark of electricity. Nucleus of it is transferred to the egg with a spark of electricity. And the purpose is what? That this spark of electricity would be making the egg cell feels fertilized, although no fertilization takes place. Now this is called as what? This is called as somatic cell nucleus transfer. Write down somatic cell nucleus transfer in bracket. Somatic cell nucleus transfer. Nucleus transfer. Clear? Now this egg is now transferred to the womb of a surrogate mother. The baby that is born is a clone of the donor. Right down. It is now transferred to the womb of. Surrogate mother. It is now transferred to the womb of surrogate mother. It is now transferred to the womb of surrogate mother. The baby that is born, the child that is born, is a clone of the donor. And clone of the donor, why? Because the nucleus belongs to the donor. The child that is born is a clone of the donor. But then it cannot be considered as exactly the same. Why? Because although it is a clone of the donor, but it won't be 100% exact. Why? Because mitochondria has not been transferred. And you know very well that DNA, we have discussed, yesterday also, that DNA is present in the mitochondria. Clear? But it won't be exactly the same. But it won't be exactly the same. It won't be exactly the same as mitochondria has not been transferred. It won't be exactly the same as mitochondria has not been transferred. Mitochondria has not been transferred. Has not been transferred. As mitochondria has not been transferred. Clear? And you know very well that DNA is also present in mitochondria. In one case, it can be exactly the same. And that is when the donor and the egg provider is the same person. Right? In one case, it would be exactly the same. In one case, it would be exactly the same. It would be exactly the same. When the donor and the egg provider, when the donor and the egg provider, donor and the egg provider, when the donor and the egg provider is the same person, when the donor and the egg provider is the same person, donor and the egg provider is the same person. In that case, it would be exactly the same. Clear? Donor and the egg provider is the same person. In that case, it would be exactly the same. Fine. So this is what? This is cloning. Egg provider is the same person. So girl child can be, but this mechanism of cloning is called as what? This mechanism of cloning is called as reproductive cloning. There is a second mechanism of cloning also, and that is called as therapeutic cloning. Right now, this mechanism of cloning is called as, reproductive cloning. This mechanism of cloning is called as reproductive cloning. Reproductive cloning. Clear? But then the other mechanism of cloning is referred to as what? It is referred to as therapeutic cloning. The other mechanism of cloning is called as, therapeutic cloning. Therapeutic cloning means medicinal cloning. Therapeutic cloning. That's called as medicinal cloning. If suppose someone suffers from disease at an old age, and for curing his disease, what would be more desirable would be embryonic stem cell, his own embryonic stem cell. Clear? Your embryonic stem cell can be used for me, my embryonic stem cell can be used for you, but your embryonic stem cell would be more adaptable for you. Clear? So for curing his own disease, embryonic stem cell would be desirable. But at this stage from where he can get, and when he was born, there was no stem cell therapy. So at this stage from where he can get his own embryonic stem cell. Now it's very easy. He would become the donor. He would become the donor. So patient himself would become the donor. And the same process would be repeated. Somatic cell nucleus transfer. And obviously this egg would be reaching the blastocyst stage. This egg would be coming under the blastocyst stage. So when it comes under the blastocyst stage, when it reaches the blastocyst stage, then we can see embryonic stem cell. Embryonic. So it is also based on SCNT technique, somatic cell nucleus transfer. So when it reaches this stage, then the embryonic stem cell of that patient can be extracted. Clear? And used for stem cell therapy. Then the concern is, then wouldn't it be unethical? Because if you are extracting a single cell from pluripotent cells from the blastocyst stage, then it would be unethical, because this would be resulting in the death of the fetus. So won't it be unethical? The answer to that is what? The answer to that is that today the doctors are debating that at what point of time it should be considered as life form. Because if suppose the one allegation is that it is responsible for destroying a life form, the second perspective is that it's also responsible for saving a life. Clear? It's also responsible for saving a life. Write down therapeutic cloning. Therapeutic cloning. Therapeutic cloning. This is for medicinal purpose. This is for medicinal purpose. Medicinal purpose. Particularly if embryonic stem cell is required, particularly if embryonic stem cell is required for a patient, embryonic stem cell is required for a patient, particularly if embryonic stem cell is required for a patient, for curing his disease, for curing his disease, curing his disease, which is not present in the stem cell bank, and which is not present in the stem cell bank due to many reasons. It can be due to the age also, because when he was born at that time there was no stem cell therapy, which is not present in the stem cell bank. The patient himself becomes the donor. The patient himself becomes the donor. Patient himself becomes the donor. Patient himself becomes the donor. Becomes the donor. The patient himself becomes the donor. And himself becomes the donor. And SCNT, that is somatic cell nucleus transfer, and SCNT is used in this case also, and SCNT is used in this case also. SCNT is used in this case also. This case also. When the egg reaches the blastocyst stage, when the egg reaches the blastocyst stage, everything is the same. When the egg reaches the blastocyst stage, and you know the blastocyst stage, embryonic stem cell of the patient, embryonic stem cell of the patient, embryonic stem cell of the patient would be extracted. Embryonic stem cell of the patient would be extracted. Would be extracted and used for therapy. Embryonic stem cell of the patient would be extracted and used for therapy. And used for therapy. Used for therapy. Clear? But this would lead to the death of the life form. Write down. But this would result in the death of the life form. But this would result in the death of a life form. In the death of a life form. Death of a life form. As such, doctors debate it. As such, doctors debate it. That at what point of time, as such, the doctors debate it, that at what point of time it should be considered as a life form. That at what point of time it should be considered as a life form. It should be considered as a life form. Life form, as it is responsible for saving a life also. As it is responsible for saving a life also. Saving a life also. Clear? So this mechanism of cloning is called as what? It is called as therapeutic cloning. So cloning can be divided into two categories. One is what? One is reproductive cloning, and second is what? Second is therapeutic cloning. Clear? And this is how SCNT technique works. Somatic cell nucleus transfer works. Now this is cloning chapter, but Remember that as far as cloning is concerned, cloning is based on somatic cell nucleus transfer. It is based on somatic cell nucleus transfer. But when you talk about triple parent baby is based on what? It is based on mitochondrial DNA transfer. See what happens. Sometimes we know that there are 20,000 genes in our cell. Out of these 20,000 genes, 37 genes are determined by mitochondrial DNA. So out of these 20,000 genes, we can see 37 genes are determined by mitochondrial DNA. So they would be also responsible for certain characteristics. Clear? They would be responsible for certain characteristic. And in the limelight of that, in the limelight of dysfunctional mitochondrial DNA, we can see that triple parent baby is resorted to. So just after the break, we'll start with what? We'll start with triple parent baby. Clear? And try to complete biotechnology today and start with disaster management.